Let us see the nucleotide excision repair pathway. So in this pathway, the crystal structure of UVRA DNA complex reveals that UVRA doesn't make direct contact with the modified thymine but does bind to DNA regions on either side of the lesion. So the first step is the formation of UVRA DNA complex. So UVRA comes to the damaged DNA and binds with the DNA. So it doesn't directly bind to the damaged nucleotide. So rather it binds either to either side of this lesion. Based on this information, it appears that UVRA makes an important contribution towards recognition of the DNA damaged lesion. This UVRA DNA complex is formed in vitro in the absence of UVRB. The recognition process appears to be more complicated in vivo, where UVRA is a part of UVRA to UVRB complex. So this UVRA DNA damage complex was formed in vitro, that is outside the cell in the lab. But if we see the formation of this complex in vivo inside the cell, so it is more complicated, and UVRA is a part of UVRA to UVRB complex. UVRB, this is another protein. It plays a central role in nucleotide excision repair by interacting with both the UVRA and UVRC, although it doesn't interact at the same time with UVRA and UVRC. It interacts with these two proteins at different times. UVRB has at least two catalytic sites that are essential for its function. UVRB combines with UVRA to form a UVRA to UVRB complex, either in the solution or on the DNA. So if two molecules of UVRA are present and one molecule of UVRB is present, they make a complex. And the presence of DNA is not compulsory. This complex can be formed either in the solution or on the DNA molecule if DNA molecule is present. Initially, this protein complex binds to DNA at some distance from the damaged site. So after the formation of this UVRA to UVRB complex, then the next step is that this complex binds to the DNA. So again, it binds to some other place other than the damaged site. It is slightly away from the damaged site. Once UVRA to UVRB DNA complex is formed, that is, in this complex, three types of substances, that UVRA, two molecules of UVRA, then UVRB, and then DNA. So this is UVRA to UVRB DNA complex. Once it is formed, the UVRB helicase catalyzes ATP dependent movement of UVRA to UVRB along the DNA until the protein complex encounters a bulky adduct or helix distortion. So when this complex UVRA to UVRB binds to DNA, it binds slightly away from the damaged site. So after binding, the UVRB helicase catalyze the movement of this complex. So this start moving along the DNA strand, and it keeps on moving until it reaches the damaged site, either a bulky adduct or in the form of a helix distortion. So it reaches on the site. The UVRA is released in an ATP dependent reaction with a concomitant conformational change in the UVRB, that produces a stable UVR-B DNA complex. So when this complex UVR-A to UVR-B complex, when it reaches the damaged site, the UVR-A is removed from this complex. So the UVR-B and DNA complex remains. So here in this figure you can see these are the two molecules of UVR-A2 and this is one molecule of UVR-B, and first this complex is formed which is UVR-A2 UVR-B, and then this DNA containing this damaged portion. So this damaged portion is present in this DNA. Then this complex binds with DNA. Look, this is the damaged site, but the complex has bind somewhere else, that is away from the damaged site. So in this way a UVR-A to UVR-B DNA complex is formed, and in the next step you can see this is the UVR-A2 UVR-B DNA complex. So it start moving, and after that this UVR-A molecules, two UVR-A molecules are removed, and now this is UVR-B DNA complex. So this is a stable complex. UVR-A functions as a molecular matchmaker in this pathway, because it has only role to match the UVR-B and the damaged site, and then it is removed from the complex. It uses energy of ATP to facilitate the formation of UVR-B DNA complex and then dissociates from the complex. So in this whole process the energy from ATP is used. UVR-C, which has a flexible linker that connects its N-terminal and C-terminal domains. It binds to UVR-B DNA complex and makes two incisions, one on each side of the lesion. So if lesion is present here, it makes two incisions, one here and one here on either side of the lesion. The first incision of the damaged strand is 4 nucleotides towards the 3' end from the lesion, and the second is 7 nucleotide towards the 5' end from the lesion. So you can see if this is the strand and this is the damaged site. So first cut is about 4 nucleotide towards 3' end, and the second nick is towards 7 nucleotides towards 5' end. So in this way two nicks are produced. This is a UVR-B DNA complex, this one, and this is UVR-C molecule. So it combines with this UVR-B DNA complex, and a complex which is UVR-B UVR-C DNA complex is formed. So this UVR-C, it introduces two nicks. The first nick here, this is 3' cut, and the second nick is here, which is 5' cut. So this UVR-C, it introduces two nicks in the strand of DNA which contained damaged nucleotide. So in the next pathway, this is UVR-D, and it keeps moving it, and then DNA polymerase 1, it repairs the excised portion, and finally DNA ligase, it ligates the ends. So in this way this pathway is completed. So how this UVR-C interacts with UVR-B and performs its function is still to be described. Nobody knows how this interaction takes place. Following 3 more steps are required to complete the repair process, and these are the UVR-D, a helicase, excises the damaged oligonucleotides, then DNA polymerase I uses the undamaged strand as a template to fill the gap, and finally DNA ligase seals the remaining nick to complete the repair process.